Hello, hi, good afternoon. Am I audible, visible? Please confirm. Okay, okay. I have submitted both the assignments and the week 3 assignment. Okay. Have you seen all the lecture videos of week 3? Okay. Okay, and I think week 4 material is also available. Okay. Any difficulty in solving the week 1, 2, and 3 till how much you have completed? Okay. Means the lecture videos are sufficient and the live interaction session is also sufficient. Okay. If you need any more help or any more suggestion, you can let me know. Please feel free. Okay. Let's wait for some time till others may also join. Then we'll start. Okay. Mm. Subhalakshmi Kumaran, son, are you there? Okay, may I know from where you are, like which college and which degree you are currently pursuing? Okay, and BSc or MSc? Okay, okay. Is this your first class you have joined? You have joined my class for the first time? Okay. Have you registered for the course, like you are solving the assignments or just learning? Sorry. Okay, means you are solving the assignments and submitting that. Have you submitted both the assignments for week 1 and week 2? 3 is currently ongoing. The deadline is open for that. You faced any difficulty in solving the assignments or were they okay for you? Okay. So if you need to discuss any specific topic, then you can let me know. We can cover that. Okay. We'll start in few minutes till I think someone also joins. Okay. 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 So I think we can start now. Okay. A short introduction about myself for those who are joining this for the first time. My name is Kajal Gachawa. I'm PMRF Research Scholar from IIT BHU, and I'm TA for this course, that is Host Pathogen Interaction. So I will be taking this course till April, and I will be helping you out in solving the assignments. Someone is there. So this is a brief introduction about me. I'm your TA. This session will be conducted on every Saturday from evening 3 to 5 p.m. And this is the week 3 live interaction session. Week 1 and 2 are already there on the YouTube. So you can visit the channel and see the video if you have not seen earlier. And the week 3 is currently going on. So I would like to the new joiners, please write your name in the chat box with your name, your course degree, and your college from where you are joining. A short introduction about you. Those people who are joining this lecture for the first time, I would request them to please write your name in the chat box. I can see there are 2, 3 new people. Please quickly introduce yourself with your name, your college, and your degree so that we can start with the assignment. Okay. Ranita from Andhra Pradesh. So you can write your introduction in the chat box. Meanwhile, we'll start the session. The lecture material for week 1, 2, 3, and 4, that is the history and development of immunology, immune organs, cells of immune system, and about the cytokines is already released on the NPTEL. So you can check it out there. And the assignments for same is also there. Today we will be solving the week 1 and week 2 assignment. So, quick revision from the last class what we have studied about. We have studied the details of history and development of immunology. We have seen the Edward Jenner story, that how he discovered that this cowpox can be helpful in protecting from the smallpox. It can be used for the vaccination. So Edward Jenner gave the first ever vaccination. So we have seen this Edward Jenner work, and Robert Koch gave the Koch postulates. There were four postulates. He have conducted one experiment using the mouse, and he have given these four postulates that how one organism can infect others. And then we have seen the Louis Pasteur experiment, in which he have shown that how weakened bacteria can stimulate immunity without causing any disease to the cells. Then we have seen this phagocytic theory of immunity from the Elie Machnikov, and serum experiment from Emil von Behring, toxin neutralization assay from Paul Ehrlich. So if you guys have any doubt in these theories, then you can let me know. Then we have seen this Nobel Prize winners of immunology, branches of immunology, and today we will be solving the week 1 assignment. So if any of you have not seen the last week's material, so you can watch it on YouTube again. And for the week 1 assignment, the first question was, which experimental approach proved that serum from animals exposed to toxins could protect other animals? So, what answer you have submitted to this? You can quickly reply in the chat box. First question for week 1 assignment was, which experimental approach proved that serum from animals exposed to toxins could protect other animals? Option B, that is the serum therapy experiments by Behring. What was there in this particular experiment? Can you summarize this very briefly in one or two sentences? Sorry, am I audible now? Is my voice okay now? I think your network is not proper, so please write in the chat box. I can't hear you. Is my voice okay now, or is it breaking, or there is echo? No, anybody else from the joiners? Can you please tell? It's still breaking. Okay, okay. Anyone else can also please confirm whether it's breaking or is it okay for all? Okay, so can you please summarize very briefly in one or two sentences what was there in the serum therapy experiments by Behring? Okay. What was the conclusion from this? That the role of antibodies is there in providing immunity. So, what is they are asking in this particular question is that which experimental approach proved that serum from animals exposed to toxins could protect other animals. So, means they are telling about the role of immunities, that is the serum. Okay, so the serum therapy experiments from Behring told this that immune cells are there. Some serum, when serum is transferred from one animal to other, it contains something. That something is the immunity. Later on, they discovered that. So the correct answer is serum therapy experiments by Behring. This is the experiment. And the next question is what is the primary purpose of the Gruber-Vidal test? We have seen this test also, Gruber-Vidal test. What was there in this test and what is the answer for this? Okay. And how we got to know this, that there were two scientists, Gruber and Durham. What they told that bacteria of similar size clump together in sera in ways specific to or determined by each serum. Okay, and Vidal used this concept for making the Gruber-Vidal test for Salmonella typhi detection. What he did that he have he developed a diagnostic agglutination test for typhoid, therefore known as the Gruber-Vidal test, for detecting Salmonella typhi present in a patient's serum. So this was his work. For a quick summary, that he have taken the patient's serum containing antibodies which contain the Salmonella typhi bacteria, and then if the patient has typhoid fever, their serum will contain antibodies against Salmonella typhi. So this bacteria will clump together or agglutinate to confirm the infection. Like there will be agglutination, it means the bacteria is present in the blood of that particular patient. So it was the first serological test to diagnose bacterial infection. So this was the Gruber-Vidal test. So the correct answer for this is to detect antibodies against Salmonella typhi in the patient's serum to diagnose typhoid fever. The next question is which of the following statements best reflect Buckner's discovery regarding alexin in blood serum? We have also seen the Buckner's experiment using alexin. So what is the correct answer for this? What was there in Buckner's experiment? Mhm. And this was the Buckner's experiment. Edward Buckner, what was his key finding? His key finding was that he identified a bacterial killing factor in serum, which he called as alexin. At that time, he gave the name alexin. So Buckner discovered that blood serum, the fluid portion of the blood without cells or clotting factor, could destroy bacteria. So he named this factor as alexin, which later known as the complement system. The complement system consists of proteins that enhance immune responses, helping antibodies and phagocytes destroy bacteria and infected cells. So he confirmed that immunity could be mediated by soluble factors in the blood. That is the humoral immunity. He gave the concept of humoral immunity. So his discovery led to the discovery of complement system. At that time, he called this particular factor as alexin. So he said that bactericidal power of blood, that blood also have bactericidal power. So what they are asking here in this question, that Buckner demonstrated that alexin could destroy bacteria independently of immune cells. Like there are no immune cells in the serum of the blood, but there are some soluble factors. Those are complement proteins or complement system. So blood also have bactericidal power. And the fourth question is what is the key observation in the fever phenomena? What was the fever phenomena? This is the fever phenomena, the immune bacteriolysis. So his experiment was that he injected Vibrio cholera into the peritoneal cavity of non-human normal animals. Then the bacteria multiplied rapidly and caused the infection. And when he injected this into other animals which were just recovered from the cholera infection, then he observed that in those patients, the bacteria swelled up, dissolved, and disappeared within 20 minutes. Why this happened so? Because the bacterial destruction occurred without any direct cellular activity. Because these organisms was just recovered from the infection, so they were immunized from Vibrio cholera. And when he injected this into the serum from immunized animal, destroyed bacteria in test tubes. Like what he did, he have taken the serum from the immune animals and added this to a test tube containing Vibrio cholera. Then also same thing happened. The bacteria were lysed in the same way, like they were swelled up, dissolved, and disappeared within 20 minutes. So this proved that bacteriolysis was due to the antibodies and complement proteins in the serum and not due to the immune cells, because he have isolated the serum from that particular immunized animals. So in the serum, no immune cells are present. That's why it is called the immune bacteriolysis, the fever phenomena. From where he also supported this immune theory, immune concept. He said that immune cells are sorry. He supported the serum theory, not the immune cells. So he said that bacteriolysis was due to antibodies and complement proteins in the serum. So this is the fever phenomena. So his observation was that Vibrio cholera undergo bacteriolysis in immune animals, swelling, dissolving, and disappearing within 20 minutes. So your next question was, which of the following concepts have contributed to the foundational theories of immunology? So out of these options, which one are correct? What we need to see here, the foundational theories of immunology, like from where immunology started. And what about the study of monoclonal antibodies? It is not supporting the foundational theories of immunology. It came very later on. Initially, scientists told that there are some soluble factors or there are some humoral factors. They were not aware of the antibodies at that time. And what about the theory of gene expression? It's not there in the immunology. And what about the this evolutionary theory? Have nothing to do with the immunology. So the correct answer is Mechnikov's phagocytic theory and Ehrlich side chain theory. And the complement system is also there, complement system theory, Behring serum therapy, and this dichotomy between cellularist and hemolarist. So these theories supported the foundational theories of immunology. So there were so many different theories. Some scientists were cellularists. They told that the chief defense of the body against infection resided in the phagocytic and digestive power of the macrophages. That is, they supported the immune cells. and some theory supported the hemolarist, like they told only the soluble substance of the blood and other body fluids could immobilize and destroy invading pathogen. Like they told there are some soluble factors or some like something like antibodies, some humoral factors are there. There were so many scientists, out of which these particular theories supported the foundational theories of immunology. And this is the very interesting question from where we have started studying the immunology. What did Edward Jenner use to develop the first successful vaccination? What he used, which virus he used for developing the vaccination? Why not smallpox virus? What he did, like what they are asking in the question. What did Edward Jenner use to develop the first successful vaccination? If they asked Edward Jenner developed for successful vaccination for what disease or using what virus? Yeah, for what virus? If it is for what virus, then you have to say it's for smallpox virus. And right now, here they are asking what he used to develop the first successful vaccination. He used cowpox virus as a vaccine for the smallpox virus. There is no confusion in these both na smallpox and cowpox. Nothing to do with rabies, influenza, polio. These are already in the side. But when they will be asking about smallpox or cowpox, how will you differentiate? Like, if how will you differentiate about Edward if you see the name Edward Jenner? First thing comes in your mind is smallpox and cowpox. How will you know that what why here the answer is cowpox, not the smallpox? How will you differentiate between these both virus related to this particular question? Because Edward Jenner work is associated with smallpox also. Yes, yes. What he used? He used cowpox virus. Okay, yes, continue. Okay. So don't get confused in this both cowpox and smallpox because they both are similar and both are associated with Edward Jenner. What is the main difference between the cellular and humoral theories of immunity? We have just seen that. What is the main difference between cellular and humoral theories of immunity? No confusion with other options like cellular theory highlighted antibodies. No, they told about the cellular means cells, nothing to do with the disease, no role of macrophages. So it is very direct and very clear that cellular theory emphasizes immune cells, while humoral theory focuses on soluble factors in the blood. And what these soluble factors were actually, those were serum, antibodies, complement, those were the soluble factors. And immune cells were all the other cells of the body. Which type of immune specificity is primarily associated with innate immunity? We have seen this in very first class, first week class. Immunity, different types of immunity. Which type of immune specificity is primarily associated with innate immunity? Why not narrow specificity? Like it is non-specific in nature, it is not specific to a particular molecular structure. It will consider all foreign particles as foreign. It will not tell that whether this is what category of foreign particle. So narrow specificity is there in the adaptive immune system where we talk about the specific immune response. But here the question is about innate immunity, which is non-specific in nature. That is the broad specificity for molecular patterns. And what about memory specificity for prior infection? In innate immunity, there is no memory. No memory cells are there for the innate immunity, or are there some memory cells? There are no memory cells for that. Memory is developed for the adaptive immune system, for the adaptive immunity, that is acquired immunity. Because this innate immunity is very short term, no? There is no memory for that. Adaptive immunity is long term immunity. So, tolerance specificity to avoid self reactivity. And, and innate immunity is non specific for that. And adaptive specificity that develops over time. So, it is the itself adaptive specificity, adaptive immunity. So the answer for this is the broad specificity for molecular patterns. Next question is, which of the following contributes to the regulation of immune response? Like for regulating an immune response, which of these are contributing? Mm. Mm. And like how, what are the justification for the chosen option? Like question is about which of the following contributes to the regulation of immune response? Mm. That will come like later on. Now, here it is the contribution to the regulation of immune response, how immune response is regulated. Others, people from the audience who have joined would like to answer or would like to join this conversation. You can have your opinion, what your thoughts about this. Think about all the options. What they are asking in this question is that which of the following contributes to the regulation of immune response? other options. So, what is the answer for this? See, the answer for this is these three. So, what is the justification behind these answers? Like how they are contributing to the regulation of immune response. We can take 5 minutes on this question, then discuss each of the options. Mm, that is okay. Sorry, what you are saying? Like if you see in that way that for the immune response, what should be there, then the antigen specific antibody production is also there, the phagocytosis is also there, like everything is there. So why only these three options are correct for this? Just give me 2 minutes. I'll clarify all the options. Let me check it once. Anybody got the thoughts on this question? Why these three options are correct and others are wrong? What is your opinion on option A and C? Mm. Here, the question is specifically about the regulation of immune response, like how immune response is regulated. Mm. Mm. Cytokines are the key regulators of an immune response. Like these are pro-inflammatory and anti-inflammatory. So like they can whether increase the immune response or they can suppress the immune response. So they are regulating the immune response. There is an immune regulation. And for positive and negative feedback mechanism, what is happening here? This is the feedback mechanism which is controlling the immune response. Like directly the regulation of immune response, what is being asked in the question. Positive and negative feedback mechanism. So what is happening here in positive and feedback mechanism? Mm. 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 So in it is giving the feedback to the immune response, like whether to upregulate the immune system or downregulate it. So it is the regulation mechanism. Same is happening with here the cytokines. Cytokines are also pro and anti-inflammatory. This pro-inflammatory may trigger an inflammation reaction and anti-inflammatory may suppress an inflammation reaction or an immune response. So the cytokines are also down regulating or up regulating and response. And same is happening here for the feedback and for the positive and feedback mechanism. It is also regulating the immune response. And what is happening here in the case of cellular interaction, such as M1 and M2 macrophage balance? What are these M1 and M2 macrophage and how they are related to the regulation of immune response? What are your thoughts on this macrophage balance? Check it out. What is written there from where you have read? You got any thoughts on macrophages? Yes. Here are two type of macrophages mentioned, M1 and M2, and they are talking about their balance. Like here it is also written the phagocytosis of pathogens. So this option is not correct here. Phagocytosis of pathogen is also done by the macrophages. But if we are only talking about phagocytosis of pathogen, then it will not contribute to the regulation of immune response. See, in option D and option H, both are talking about macrophages only. But there is a difference. Phagocytosis is related to inflammation. I don't think so. It's directly related to inflammation. Phagocytosis is removal of something from there which is not required. If you have any thoughts, you have studied it somewhere, then let me know. Otherwise, also let me know if you are not aware of this. I will clear this. Mm. So, macrophages are of two types, M1 and M2. What are these M1 macrophages? Like same thing happening here, we have just discussed in the case of cytokines, pro and anti. So the M1 macrophages, what they do, they generate a pro-inflammatory response. Pro-inflammatory response means they are responsible for the killing of microbes. Like any pathogens are there, they are responsible for killing that. And what is happening in M2 macrophages? M2 macrophages are used for that anti-inflammatory response. Anti-inflammatory means they are helping in the tissue repair, wound healing, and phagocytosis of apoptotic cells, like the dead cells, they are removing from that. So these M2 macrophages are removing things which are not required. And this M1 macrophages, what they are doing, they are killing those cells. Like there should be balance between this both the cells. See in here what happening, what is they are saying cellular interactions such as M1 and M2 macrophage balance. So macrophages are promoting inflammation and pathogen clearance for this M1, and for this M2 they are supporting tissue repair and resolving the inflammation. So there are two different types. So there should be balance between these macrophages. If only M1 is there, like all the macrophages will be promoting inflammation and pathogen clearance. Like they will be clearing out the pathogen and promoting the inflammation. So there will it will be a not good idea. If there are only M2, no M1. So only M2 will what they will do? They will repair the tissue and resolve the inflammation. So there should be balance between promoting inflammation and repairing inflammation, like killing the cells and removing those cells using phagocytosis. So there should be balance between these both. So if we are talking about the balance, it is coming in the regulation of immune response. If we are talking only about the phagocytosis power of pathogens or the triggering of inflammation using macrophages, then that will not come in the regulation of immune response. So you got the answer now, or any more doubt regarding this? There are two different types of macrophages. What we know about macrophages generally when we see the name macrophages, first thing comes in our mind is phagocytosis. Macrophages do phagocytosis, that's it. But there are different types of macrophages. Like macrophages, there are different stages of macrophages. So have a short note on this, write 2 points about this, that M1 macrophages are promoting inflammation and pathogen clearance. And this M2 macrophages are supporting tissue repair and resolving inflammation. Got the point? Okay. Others also, is this point clear, or you have any doubt regarding this, or you have any suggestion? Others can also join if anybody wants to. And what about other options? Why not this pattern recognition receptor in adaptive immunity? Why this is not involved in regulation of immune response? Mm. And they can also regulate the immune response. What is the role of pattern recognition receptor? What they do? Your voice is breaking. Can you please repeat what you said right now? Your voice is breaking. You can write in the chat box if problem with the internet. Yes, 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 you are audible now. Mm. Mm. So what they do, pattern recognition receptors, they recognize the pathogen, like they detect the pathogen and initiate an immune response. Like they can initiate an immune response. They are involved in an immune response. But if we talk about the regulation of immune response, like we have seen here in these three options, regulation means upregulating or downregulating, promoting or suppressing. There should be both ways. So, pattern recognition receptor can initiate an immune response by recognizing the pathogens, but they cannot regulate an immune response. They are not directly involved in the regulation of immune response. That's why this is not the correct option. You got my point? And what about other options like complement activation pathway? Why complement activation pathway is not involved in the regulation of immune response. Mm. It is a response, like it enhances the immune response by promoting opsonization, inflammation, and pathogen lysis. But it is not a mechanism of regulation. It is like promoting the immune response. So next is the role of bacteria in vaccine development. There is no direct regulatory role of bacteria in vaccine development to the immune response. It is not involved in the regulation of immune response. And what about antigen specific antibody production? Just a minute, give me a minute. What about antigen specific antibody production? Hmm. Hmm. It is not a regulation mechanism. And same thing with the phagocytosis of pathogen. We have seen this in the case of macrophages, like it is the response to the pathogen. It is not a regulation mechanism. So now I hope all the options are clear, whether they are correct and whether they are wrong. What is the reason behind that? Like you don't need to mug up the options like whether it is this or this. You should know the reason behind that. So if you still have any doubt or something is not clear, let me know, we can discuss it. And for other people also who have joined, are these questions clear to you or you have some doubt regarding this? We can discuss it more if these are not clear. Please confirm in the chat box whether it's clear, and if it's not clear, then also please confirm in the chat box so that we can proceed. Those who have joined, please confirm in the chat box. Are these questions clear to all of you? Okay, then let's see next question. Whose work led to the development of the first vaccine? The favorite question of immunology. Is the story clear now? Edward Jenner's story. Is it interesting? Okay. So here ends the week one, assignment one. So if any question is not clear to you, we can take it again. Otherwise, we can move to week two, quiz two. We can check one by one all the questions. Like, is this questions clear? Otherwise, you can stop me here. We can discuss it again. This is about agglutination, Vidal test from Buckner, this Alexin from Buckner, Pfeiffer phenomena, and this all the foundational theories of immunology for successful vaccination. No confusion in this question, na? Smallpox or cowpox. They used cowpox virus for developing a vaccine for the smallpox. So remember this line. And the cellular and humoral theories of immunity and the innate immunity, basic, and this detailed question about regulation of immune response, and famous and most interesting question, Edward Jenner. So let's move to week two assignment. So first question was, what condition highlights the consequences of lymphatic blockage due to a parasitic infection? What is the answer for this? Okay, why and how? What is the question? Consequences of lymphatic blockage due to parasitic infection. Then, so you know the story, what is happening here in the case of elephantiasis, how the disease is spreading and what is happening in this. Are you aware of the story or you want me to explain this in detail? Okay. So this elephantiasis is a disease. It looked like this, like one of the body part or the organ, it starts swelling up very dangerously. So elephantiasis is also known as lymphatic filariasis. Why it is called lymphatic filariasis? Because it is a parasitic infection that leads to the blockage of lymphatic vessels. Somebody raised hand. Anybody want to say something? I think somebody raised hand. Shubha Lakshmi. Like it's not visible clearly for others. Is it visible clearly or like what is the problem? Is it small or blur or what? Others, please confirm whether it is blur to all. Is someone other also please confirm? Okay, we are waiting. You can join again. Okay, okay. It's clear for others. Okay, might be some network issue. It's clear now. Okay. So it went blur just now only, or from starting you are seeing a blur presentation? Lakshmi, from starting all the slides you have seen in blur quality, or just now only it went blur? Okay, okay. I thought from starting also you are seeing the blur presentation and you don't understand. Okay. Okay. I remember in last class also slides were initial 5 6 slides were not visible and everybody understood all the concepts without seeing the story slides. So at any point you can stop me or you can cross check. Okay, so let's see this lymphatic filariasis. So it is a parasitic infection that leads to the blockage of lymphatic vessels. This blockage results in severe swelling, that is the lymphedema, typically in legs, arms, genitalia due to fluid accumulation. This condition is primarily caused by filarial worms. The names are Wuchereria bancrofti and Brugueria malai and Brugueria timori, which are transmitted through mosquito bites. These mosquitoes are Anopheles, Culex, Aedes, and Mansonia species. So, what is happening here? See the life cycle of lymphatic filariasis. This have life cycle, half of life cycle in the mosquito and half of life cycle in the human. So what is happening here? When an infected mosquito deposits larva on the skin while biting and the larva enter the wound. Mosquito have a larva, it will bite on our human skin and the larva will enter our bone. Then this larva migrate to the lymphatic system where they grow, mate and form nest. The nest causes blockage resulting in swelling and fever. Like this larva enters inside our body and it start multiplying and blocking our lymphatic system. So in adult, what happened? This female worms start producing microscopic worms called microfilariae, and these swarm in the blood at night when mosquito bites. So these worms will again, lymphatic system is spread all over our body. So it will spread to everywhere. And at night, what happens? It reaches to our this area, and again some other mosquito bites us. Like a mosquito bites the infected person, ingesting the microfilaria along with the blood. Like this, female worms were multiplying in our human body, in our blood, and they were producing this microscopic worms called microfilaria. These are again producing small more filari. And when the mosquito bites the infected person, then it ingests the microfilari along with the blood. Like it suck up the microfilari from our blood. Then this microfilari develop into larva over a period of week. In mosquito, in the body of mosquito, this microfilari again develop into a larva in some time. Then again, this mosquito containing larva in his blood, it will bite to the human. It will place the larva here on the human skin. Then again, it will enter in human. Then again, this female worm will produce microfilari, and it all the time it will go and block our lymphatic system. So when lymphatic system is blocked at particular place, then it will swell up our, like whether it's legs, arms, or genitalia. It will swell up and the fluid accumulation will occur. So this condition will take place. So half life cycle is taking place inside mosquito and half is taking place in human. In mosquito, what is happening? Microfilaria is developing into larva. And in human, what is happening? This larva is developing into worms. And these worms are, female worms are again producing microfilaria, and it is again transmitted into mosquito. So these are the families of mosquito which are responsible for transmitting this disease from infected person to healthy person. And these are the names of the this worms which are responsible for causing this condition. And every time they will go and block the lymphatic system. So this condition will take place, and when a leg or a arm is swelling up like this, like an elephant, so this disease is called as elephantiasis. So this is the story of elephantiasis or lymphatic filariasis. So is it clear? Okay, for others also the story is clear, like what is happening here. They may ask question in some other way also, like they will not ask what is happening during the lymphatic blockage, like it this disease the lymphatic blockage is happening. They can ask what is happening in lymphatic, this elephantiasis, and why lymphatic blockage is happening, what is the life cycle, like how in mosquito what is happening, in human what is happening. So you should know the complete story of elephantiasis. So is it clear for others also? Please quickly confirm. Okay, like you all already knew this story, what is happening, how lymphatic blockage is happening, or just mugged up what is happening, like in which condition lymphatic blockage is happening, or the story you were already aware. Mhm. Worms. Okay. Because there are this worm have life cycle in both mosquito and both the human. It completes its life cycle in two different hosts. And these mosquitoes are the transmitter or the transporter or the vehicles for transmission of this disease. So you might be thinking that this mosquito are responsible for causing the disease. No, the causative agent is this worm, these worms. The transporter, transmitter, or the vehicle for this disease are the mosquitoes. So don't get confused between elephantiasis is caused by these mosquitoes or by this worm. The worm is the causative agent. Okay, let's move to next question. Mucosal surfaces play a pivotal role in immunity due to their unique structural and functional characteristics. Which of the following best explains their immunological significance? What is the answer for this? For the mucosal surfaces, what are their immunological significance? Mm, mm, mm. So the answer for this is they are enriched with specialized immune cells and secretory mechanism, forming a critical barrier to pathogen entry across extensive internal and external surfaces. So what is happening here? The route of infection for pathogens are like uh, that can be entered in human body through mouth or respiratory tract or from the gastrointestinal tract and from the female urogenital tract or the male urogenital tract. These are the route of infection for pathogens, and mucosal surfaces are present at all these places. So what is happening here? The mucosal surfaces, like see this mucosal surfaces, that is the respiratory, gastrointestinal, and urogenital tracts, serve as primary entry points for pathogens, making them crucial for immune defense. So their structural and functional adaptations include the MALT, that is the mucosal associated lymphoid tissue. It includes GALT, that is the gut associated lymphoid tissue, and BALT, that is the bronchus associated lymphoid tissue. So, and these contains the dendritic cells, macrophages, T cells, B cells for immune surveillance. Like these tissues contain the immune cells. And the Bayer patches and tonsils detect pathogens in gut and the airways. Like they will detect the pathogen and stop their entry there itself. They will not allow the pathogen to enter into human body. And they also have the secretory mechanism, like they produce mucus, which are produced by the goblet cells. And this mucus, what this mucus do, this mucus traps the microbes. And they also produce the secretory IgA, which neutralize pathogens without triggering any kind of inflammation. And they also secrete some of the antimicrobial peptides, that is the defensins, and some enzymes like lysozyme, which break down bacterial cell walls. Like they secrete some molecules. What are these molecules? They secrete the mucus. Mucus is produced where? In the respiratory tract, the mucus is produced. And we can see from our nose, our throat, the mucus is produced. The mucus is coming out that traps the microbes. So that will not allow the pathogen entry. And in my antimicrobial peptide, what is happening there? Like we have our tears, our saliva that contains these enzymes, lysozymes. So if pathogen is trying to enter from your eyes or your mouth, then your tears and saliva will break down the bacterial cell wall and it will kill there itself. And it can also secrete some IgA, which neutralize the pathogen. So, like first of all, here there are some immune cells and physical barrier. It is stopping the entry of pathogen. Then also, if pathogen trying to enter, then it have some secretory mechanism which will trap or kill the pathogen, and it also have the epithelial barrier function. Like the tight junctions prevent microbial invasion that will not allow the entry of microbes, and the M cells in the gut transport antigens to immune cells. So, if by chance it enters in the gut by bypassing any of the pathway or like any of the this path, then M cells in the gut, they will transport this antigen to the immune cells. Further immune cells will start killing off those pathogens. So the mucosal immunity is highly specialized to prevent pathogen invasion while avoiding unnecessary inflammation that could damage delicate tissues. So, what is happening? What was asked in the question? They asked mucosal surfaces play a pivotal role in immunity due to their unique structural and functional characteristics. So, which of these explain their immunological significance? So, what is the answer for this? Correct answer is like they are enriched with specialized immune cells and they have the secretory mechanism, which forms a critical barrier to pathogen entry across extensive internal and external surfaces. So is this clear? There are three major roles of this mucosal surfaces. Okay, like they have immune cells, they have some secretory mechanism, and they also have this epithelial barrier. So the next question is, what is the significance of using irradiated mice in immune research? What are these irradiated mice? Okay, like we suppress or like remove their own immune capacity. Then we can check our external immune capacity whether it is working or not. So there is a small transfer experiment. You all might have seen this in the video lecture. I will briefly explain this. Is this clear? Anybody want to explain this first? Like you all might have seen the video. So first of all, I would like to know from your side what you understood from this transfer experiment. What are your thoughts on this? Anybody? Mhm. Mm. Mm. Anybody wants to add on this third case, what is happening there? Like first, one and two, you explained that is okay, that is correct. What is happening in third case? Anybody would like to add on your thoughts on this? Anybody else who have seen this video or understood this experiment very well would like to add their thoughts on this third case, what is happening here? No problem if you will speak wrong, no problem. I will carry on. I will continue that. I will explain whole experiment again. But first of all, I would like you guys to add the thoughts. Like Shweta explained this first and second case, that is absolutely correct. What is happening in the third case? Anybody? Shubha Lakshmi, Pranita, Kirti? No? If it is not clear to you, then also let me know. If you want me to explain, then also let me know. I will explain this experiment. Yes, no, anything, speak up. It's not clear? Okay, no problem. I will take it. I will explain this. So see here what they did in a transfer experiment. Transfer experiment is done to investigate a particular cell type, play a crucial role in defense against virus infection, to know the role of immune cells. So in irradiated mice, what we do, we give a high dose of irradiation to the mice so that their native original immune system will stop working. Like we are removing their original immune capacity from the mice. Okay. So what we have done in this experiment, we have taken a group of syngenic mice, like all similar mice we have taken. In the first case, this black one are the normal mice. They have their own immune ability or their own immune capacity. We have not done anything with their immune system. These are absolutely normal mice. We have infected these mice with the virus. Then after some time after the infection, these mice had their immune system working, functioning well. So they developed some immune response towards this virus. Like now they have some immune cells in them. So we isolated the discovered cells. Like the newly formed immune cells, we have isolated from this mice. So this is the part one. Normal mice, no irradiation given, well functioning immune system, infected with the virus, they developed an immune response. Then we have isolated their immune cells. Okay, this part one was there. In part two, what we have done, we have taken this irradiated mice. Like these mice are these mice are irradiated. They don't have their own immune capacity. So what we have done here, we have irradiated them, no immune capacity. Then we have transferred the discovered cells after infection, like these discovered cells, which we have initially isolated from this particular case, this virus infected case. We have transferred these immune cells into this mice. Like this mice don't have its original immune system in its own body, but it got the immune cells from this case. We have transferred immune developed cells. What happened here? This mice survived. This mice is protected from the infection, no viral infection. In second case, what we have done, we have taken the same immune irradiated mice, like without any immune system working. Then we have transferred cells after without, then we have transferred discovered cells after without infection. Like we have transferred the cells from some other host or like some other animal, but that animal was not initially infected with virus. Here we have infected this mice with virus. Like its immune system is activated. It have produced the immune cells. These cells are transferred into this mice. So it received the immune cells from this case, but in this we have transferred the immune cells from some other mice which was not originally infected with virus. Like it did not develop any type of immune response or antibody towards that virus. So we have transferred these cells and this mice did not survive. Like this mice died. You understood what is happening here in this case. Is it clear now to others as well? Like these mice did not have any immune cells transferred from other the virus infected cells, transferred the immune cells but from a normal mice without antibodies from the virus. So it died. It did not survive. And this particular mice got the immune cells from the virus infected cells, like it contained the immune cells. So this mice survived. So this is happening here in this case. Clear to all of you? Let's move to next question. So this is the detail of this experiment, like why we are using this immune irradiated mice. So I will quickly take this. So the irradiated mice are commonly used in immunological studies because exposure of high dose radiation destroy their bone marrow and immune cells. So they will become immune deficient. So what applications we can use, like where we can use those mice, immune irradiated mice, we can use that in bone marrow transplantation studies, in adaptive immune therapy research, and in xenotransplantation studies, and infection and virus research. Like we can transfer immune cells from outside, from externally to those mice, and we can see whether what is the response of that, because those mice will not have their own immune cells. So what they asked in this question, that what is the significance of using irradiated mice in immune research? So the significance is to eliminate their immune capacity, allowing for the testing of new immune therapies. So this was the transfer experiment. And the next question is, what process ensure that only non-self-reactive T cells survive during development in the thymus? What is happening here? What they are asking, what process ensure that only non-self-reacting T cells survive during development in the thymus? Okay, very good. Other also for everybody, is this clear or you want me to discuss a detail of this? Okay, because it's direct question, so I don't want to take the details of this. But still, if it is not clear to you, the behind the concept is not clear to you, then you can let me know. I will quickly take that. For all of you, this is clear, na? This selection procedure in thymus, T cell selection. Okay. Okay. So this is the process of T cell selection in the thymus. I will be sharing this PPT with you guys. So the details are mentioned here. So you can, it is very simply written. I myself have written this. So you can just read and understand this. If it's not clear, if you have any doubt, then ask me in the next class. We can take it again. Because we are running out of time and my laptop battery is also dying. There is no power since morning. So I want to quickly finish this. So the next question is, which of the following are functions of lymphatic system? See these options and quickly give me the reason behind all of the options if it is clear to you. Which of the following are functions of lymphatic system? They are talking about the lymphatic system. What are their functions? Mm, mm, mm. And what about other options like hmm, mm-hmm, mm, mm, mm. Okay, so what is happening here? The first option is like reclaiming excess interstitial fluid to maintain fluid homeostasis. It is clear, we just discussed this. And for the facilitating antigen presentation in lymph node to activate immune response, yes, this is the main function of the lymphatic system itself. What we are studying in the already in the organs of immune system, that the antigen presentation in lymph node will be done there, and for producing red blood cells in response to low oxygen level. No, in lymphatic system, the RBCs are not produced. Where are RBCs produced? In bone marrow, the RBCs are produced. And absorbing dietary fats from the intestine. How? Anybody? Anybody else know about this? Like absorbing dietary fats from the intestine. This option, have you studied somewhere in the lymphatic system? Anybody have a thought on this? There are some specialized lymphatic capillaries known as the lecteals. Have you heard this term lecteals? Those are located in the small intestine. They absorb these dietary lipids or dietary fats and transport them via the lymphatic system. Like in small intestine, there are some capillaries called lecteals. Those lecteals absorb the dietary lipids and transport them via the lymphatic system. So this absorbing dietary fats from the intestine, from the small intestine, it is a function of lymphatic system. That's why this option is correct. And next one is removing carbon dioxide from the bloodstream. Is this the function of lymphatic system? Removing CO2 from the bloodstream. Okay. And preventing tissue swelling through the drainage of excess fluid. Yes, this is a function of lymphatic system. Producing hormones to regulate the metabolism. Lymphatic system don't produce hormones. Hormones are produced where? In the endocrine system. And synthesizing antibodies directly in lymph nodes. No, antibodies are not synthesized there. And this is not the function of the lymphatic system. So this question is also clear. This reasoning for all the correct options and the wrong options I have mentioned in the slides. So you can check it out there. Next question is, which of the following statements best describe the role of M cells in the immune system? What are these M cells and what is their role in the immune system? Option C. Why? What is the reason behind this? The justification for this? Hmm. So this is the role of M cells in immune system. Is this question clear to all other people? What is happening in the M cells? They are the specialized epithelial cells found in the MALT, particularly in Peyer's patch of the small intestine. Their primary function is to transport antigen and pathogen from the intestinal lumen to the immune cells, facilitating immune surveillance. So, how this M cells function? They transcytosis of antigens. These M cells capture antigen, bacteria, viruses from the gut lumen, and then they transport these molecules across the epithelial barrier via vesicular transport, and the antigens are delivered to dendritic cells, macrophages, and lymphocytes in the Bayer patch, like the transcytosis of the antigens. Then what they do, they initiate the mucosal immune response by, like, once antigen reaches immune cells, T and B cells are activated, leading to the targeted immune response. And B cells may later differentiate into plasma cells that produce IgA, a key antibody in mucosal immunity. So their role in the immune system is like for the transcytosis of antigens and the initiation of mucosal immune response. So what was the question? Which best describes the role of M cells in the immune system? So M cells facilitate the transcytosis of antigen and pathogens across the epithelial barrier to immune cells in pair patches. So this is the correct answer for this. Next question is, which of the following cells is primarily responsible for antigen phagocytosis in the skin? Which cells are responsible for phagocyting antigen in the skin? Langerhans cells. What are these cells? Like, these are the specialized antigen presenting cells found in the epidermis of the skin. They play a key role in phagocytosing antigens, processing them, and presenting them to the T cells to initiate an immune response. What are their functions? Like the functions of Langerhans cells is the phagocytosis of pathogens and antigens. These Langerhans cells capture and engulf microbes, allergens, and foreign particles that penetrate the skin barrier. Like they phagocytose this antigen and pathogen. Then what they do, they present this like antigen processing and presentation. They migrate to lymph nodes and present processed antigens via MHC class II molecules to naive T cells, triggering adaptive immunity. Then they do the immune surveillance. They constantly monitor the skin for potential threats and play a role in cutaneous immunity. Like we have seen here in the case of this, there is a mucosal immunity, like how mucosal cells are helping in providing the immunity. They are secreting this IgA antibody and by transcytosis of the antigen. This is happening with the help of M cells. It is mucosal immunity. And here in the case of uh, this antigen phagocytosis of the skin, like Langerhans cells, M cells are responsible for mucosal immunity by transcytosis of the antigen and producing IgA. And Langerhans cells, they are responsible for the cutaneous immunity by they are they are capturing the antigen, processing it, and presenting it to MHC class II molecule. So you should be knowing about this both the cells in the case of providing immunity, like M cells and Langerhans cells. Is this question clear, na? Langerhans cells, the role of Langerhans cells. Oh, okay. So the okay. So the next question is, if a knockout gene affects embryo development in the animal model and results in lethality, which strategy for gene knockout would be useful? What they are asking in the question. Okay, conditional knockout. What is like knockout? What they want to ask in this question. Okay, okay. So in this case, what they are doing, if a knockout gene affects embryo development in the animal model and results in lethality, which strategy for gene knockout could be useful? Hmm. Okay. So why conditional knockout? Why not conventional knockout? Hmm. 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 So what is happening here? In the case of conventional knockout, like your target gene is knocked out in all tissues at all the times. In conditional knockout, what is happening? We can control where and when your target gene is knocked out, like at what tissue, at what organ this should be knocked out, and for what duration, like when it should be knocked out for the whole organism or for a specific place, specific time it should be knocked out. This is happening in the conditional knockout. So in question, what they want to know, like if a knockout gene affects embryo development in the animal model and results in lethality, like we want specifically at the embryo development. So we will use this conditional knockout. If we use this conventional knockout, then it will completely knock that gene out for the entire duration of that animal model, not only at the embryo development. So don't get confused between this conditional and conventional. Is the difference clear? Okay. Next question is, in the context of stem cells, which of the following statements are correct? Like there are different type of stem cells. Hmm. 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 Like, is the difference clear between all these different types of stem cells, or is it confusing for you, like the totipotent, pluripotent, unipotent, multipotent? Is this difference clear, or like anybody want to summarize quickly what are these different type of cells? Hmm. Hmm. Yes. Hmm. 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 Not all. So for others also, are the difference between these different type of cells clear, or you want me to discuss totipotent, pluripotent, unipotent, and the multipotent stem cells? Here, what is happening? The transgenic mice can be developed from pluripotent stem cells. Yes, this can be developed. Pluripotent stem cells can give rise to all cell types, including placental cells. No, this is the function of the totipotent stem cells that can develop an whole organism, including the placenta. Pluripotent stem cells can form all type of cells, but excluding placental cells. So this option is wrong. The unipotent stem cells are restricted to differentiate into only one cell type. Yes, from the name also we can see that unipotent. So it will differentiate into only one cell type. And the embryonic stem cells are used to develop knockout mice. Yes, embryonic stem cells are not used to develop transgenic mice. No, we can develop using the embryonic stem cells. Multipotent stem cells can't generate biochemically related cell types. No, it can generate. Totipotent stem cells can't develop the placenta. No, totipotent stem cells can form both the organism as well as the placenta. So these are the different cell types that the definition should be clear to all of you. Don't get confused between these type of cells: totipotent, pluripotent, multipotent, and the unipotent cells. So this is the last question of week 2. Which of the following statement about lymph is true? Lymph is rich in antigens that originate from the tissue. So this is the very direct question. So here we complete the week 1 and week 2 questions. So this was today was the basically assignment question because we have 2 weeks assignment to solve. And for the third week, the topic I think is cells of immune system, and for the fourth week, that is the cytokines. So have you guys seen the video lectures from the YouTube channel? No problem. We will discuss the questions in next week. Like next Saturday, we will take the third week assignment and also discuss some concepts because we have only 10 questions to do in the next week. So we will be okay. 7 p.m. 7 p.m. session is not mine. My session is for Saturday 3 to 5 p.m. 7 p.m. session is will be taken by some other TA of this course. Like you might have taken the last Saturday course from that TA who's taking at 7 to 9 p.m. Anybody attended the 7 to 9 p.m. session for the Saturday? You can check it out. Like for this particular course, there are, I think, two or three TA are taking this course. So there will be one more live interactive session from some other TA from 7 to 9 p.m. You can join that also. But that is not from me. Written session from all the TA, then explore the thoughts of all the people. Like everybody's way of explaining is different, everybody's thought is different, everybody's way of teaching is different. So you can attend and explore. Any, okay, any other doubt or any other suggestion, any other comment? Was this class helpful for you? Was spending these 2 hours helpful, worthful? Okay, you got to learn something new, or were you aware of all these things earlier? Okay, one by one, you can give your feedback. It is very valuable for me for spending my 2, 3, 4 hours on this. One line feedback if you are interested to give. Most welcome. That means a lot. For other people who have joined for the first time, do you have any feedback for this session, for this class? Okay, thank you. Means a lot. Okay, I try to explain it in very simpler way, like how I understand these things for myself. I try to deliver in that way. Okay, thank you for the suggestion. Anybody else? Welcome, thank you. Any other feedback, comment, suggestion? This PPT will be shared to you. Like you got the presentation and the video lecture for last 2 week. Have you seen that? Have you seen the video lectures of week 1 and 2 for this session? And the PPT presentation I have also shared. You guys got that? You got the PPT, but video lecture link to the YouTube channel. Okay, okay. So you can check it out there. Those people who are attending for the first time, you will get this presentation and the video lecture shared with you. So you can check it out there. If you have any suggestion, any comment, please write it down. We will discuss in the next Saturday class from the same time, 3 to 5 p.m. Okay. Thank you guys. Thank you for joining. Wills. Okay. Thank you. Okay. Thank you. See you on the next Saturday. Bye bye. You can please leave for the class. It is over. Okay.